Hi everyone, my name is Faiza Baharum and I am a postdoctoral fellow in the CEDAR lab at the NIH Vaccine Research Center. Today I'd like to share with you a recent paper that we published in Nature Immunology entitled Intravenous Nanoparticle Vaccination Generates Stem-like TCF1 positive neoantigen-specific CD8 T cells. There is a growing appreciation for the role of the immune system in fighting cancer. We now know that T cells are important in mediating tumor regression based on the success of checkpoint inhibitors. These are therapeutic antibodies against inhibitory receptors such as PD-1 that can enhance function of T cells. Some of these T cells target cancer neoantigens, a class of MHC-bound peptides that arise from tumor-specific mutations. Indeed, different cancers have different degrees of somatic mutations, and this seems to positively correlate with the response to checkpoint inhibitors. Together, this underscores the importance of T cell breadth in mediating anti-tumor efficacy. And so, with advances in next-generation sequencing, we can now identify mutations in each individual patient's tumors. Algorithms can then predict the binding of these epitopes to MHC molecules for presentation to T cells. Cancer vaccines are designed to use this information to amplify both the magnitude and breadth of T cell responses. And this can be done using various platforms, but in the lab we have focused on a peptide-based vaccine that is delivered with an adjuvant. Take a second to introduce our vaccine platform that we call the self-assembling nanoparticle TLR78 agonist, or SNP78A for short. This is work that we had previously published in Nature Biotechnology. Briefly, the CEDAR lab developed a vaccine platform that standardizes the delivery of any given neoantigen by directly conjugating the antigen to an adjuvant. In this case, we've used a TLR78 agonist. This is done by chemically modifying the peptide that contains the desired neoantigen, allowing it to self-assemble into nanoparticles that can be taken up by dendritic cells. We showed in the Nature Biotech paper that SNP78A increases both the magnitude and breadth of neoantigen-specific CD8 T cells compared to conventional vaccine strategies such as using free peptide and poly-IC. So the focus of the current paper is to assess whether we can optimize T cell immunity by modifying the route of vaccination. For this, we used our vaccine containing REPS1, which is a neoantigen expressed by MC38, a colon carcinoma cell line. And this has been mass spec validated to be expressed on the surface of the tumors. We used a prime boost regimen of 2 weeks apart and collected blood to measure the immunogenicity of those responses based on tetramer staining. Comparing sub-Q and IV vaccination, we can see that sub-Q gives a 10-fold higher response compared to IV at the same dose. 2 weeks after the boost, we challenged the mice with tumors by implanting MC38 in the subcutaneous flank. Some groups of those animals also received checkpoint inhibitor at the time of challenge to assess if that can be synergistic effects. As you can see in this tumor growth curve, the unvaccinated or naive animals here in black grow large tumors regardless of whether they received checkpoint inhibitor, in this case it's anti-PD-L1, or not. In blue, we see that the sub-Q vaccinated animals with their high magnitude responses were able to control the tumor growth. At 10-fold lower responses, IV vaccinated animals could not control the tumor unless they were provided with checkpoint inhibitor. And so we wondered, why are the neoantigen-specific CD8 T cells generated by SNP-IV responsive to anti-PD-L1? To answer that question, we looked into the literature where there has been a great effort to understand the heterogeneity of exhausted CD8 T cells and how they respond to checkpoint inhibitor, especially using chronic LCMV models. As it turns out that PD-1 expressing CD8 T cells can be subdivided into stem-like or terminally differentiated effectors based on the expression of the transcription factor TCF1. So by blocking the PD-1, PD-L1 signaling axis, the stem-like cells can give rise to more stem-like cells, and those cells can self-renew, or they can give rise to terminally differentiated effector cells that can go on to kill tumor cells. So we wanted to know if we can find the subset of stem-like cells within our tetramer-positive CD8 T cells. In the spleen, we can detect a higher proportion of TCF1 positive cells following IV vaccination compared to sub-Q vaccination. We then asked the question, do sub-Q and IV vaccination generate CD8 T cells with different transcriptional programs? So to better understand the differences at the transcriptome level, we sorted tetramer-positive CD8 T cells from the spleens of mice and performed single-cell RNA sequencing using the 10x genomics platform. And based on their differential gene expression, we could identify 12 clusters that are visualized here in this UMAP. And if we break it down to cells from IV vaccinated animals versus sub-Q vaccinated animals, we see that the cells from IV animals cluster in this bottom left corner, whereas cells from sub-Q vaccinated animals are mostly represented in this top right corner. Of the top differentially expressed genes, we see stem-like genes such as TCF7 that encodes for TCF1 come up for these red and pink clusters, whereas effector genes such as granzyme B are highly expressed in these blues and purple clusters up here in the top right. So when we summarize that per animal, we can again show that a higher proportion of tetramer-positive cells in IV animals are stem-like cells, whereas a larger proportion of tetramer-positive cells in sub-Q animals are effector cells. As these cells are observed in non-tumor-bearing animals, we wanted to know if the stem-like cells can also be beneficial in the therapeutic setting. So here, we implanted tumors on day 0, and then we treated them and the tumors are palpable on day 7. They received a boost on day 14 together with a checkpoint inhibitor. In this experiment, the dose of the IV vaccination was increased to 32 nanomoles to ensure more similar magnitude of the CD8 T cell response. So if we look at the tumor growth curve, again, the tumors grow out in naive unvaccinated animals here in black. Surprisingly, sub-Q vaccination, here in blue, despite the 10% responses, was insufficient to control tumor growth. However, IV vaccinated animals were able to mediate tumor regression. And we know that this is antigen-specific and CD8 T cell mediated because vaccinating with an irrelevant antigen was insufficient for tumor control. And so at this point, we were wondering if there may be differences in the infiltration of CD8 T cells into the tumors. So we collected spleens and tumors and measured the tetramer-positive CD8 T cells. The numbers of tetramer-positive CD8 T cells after sub-Q here in blue or IV vaccination here in red were comparable on both sides. And if we focus on the spleen, we again see that in the therapeutic setting now, there are more stem-like cells, so TCF1 positive, PD1 positive cells in the spleen after IV vaccination compared to sub-Q vaccination. So we wanted to know whether these stem-like cells are responding to anti-PD-L1 by generating more effectors. To answer that question, we broke it down to its parts to see how the IV vaccination is working. So here I'm showing you the tumor growth curve, the survival curve, and the magnitude of responses. If we only prime once, or prime once with anti-PD-L1, or if we prime and boost without anti-PD-L1, we see that there is only some tumor regression and a slight improvement in survival. However, by providing anti-PD-L1 after prime and boost here in red, we get much better tumor control and survival. And if you look at the magnitude of the responses, we see that we now have 6-fold higher numbers of CD8 T cells, tetramer-positive CD8 T cells. And by assessing TCF1 and granzyme B expression in these tetramer-positive cells, we show that we not only have higher numbers of stem-like cells a week after that boost, as shown here in the red line, we also have a huge expansion of effector cells. So to summarize, I've shown you that modifying the route of vaccination affects T cell quality, and having stem-like cells is beneficial as they can expand upon checkpoint inhibitor treatment. But now we want to understand the mechanism of how this is happening. And so we revisited the single-cell RNA-seq data and reconstructed a developmental trajectory for these tetramer-positive cells using monocle 3. Briefly, how that works is that we can order cells by their progress through differentiation by maximizing the transcriptional similarities between successive pairs. So what we see here in this pseudo-time trajectory is that the stem-like cells appear to be less differentiated than effector cells. And this brings us to the next question. Are the CD8 T cells generated by IV vaccination less differentiated due to the differences in antigen presentation? And so, using fluorescently labeled vaccines, we could track the pharmacokinetics of the vaccine. Here we're looking at confocal images of the vaccine draining lymph nodes after sub-Q vaccination. And in the bottom row, here in white, you can see that the vaccine is detectable in the lymph node up to 2 weeks later. And in fact, by flow cytometry, we can also measure it up to 3 weeks later. In contrast, IV vaccination results in a more transient systemic distribution of the vaccine. Looking at the different organs by flow cytometry, and this is at 6 hours after immunization, we can detect the vaccine primarily in the spleen, but also some of it in the liver and the lungs. Within the spleen, there is a peak at 6 hours where there are a lot of monocyte-derived dendritic cells that are vaccine positive, followed by macrophages and conventional dendritic cells. So, to assess whether monocyte-derived dendritic cells are indeed important for vaccine responses, we compared them in knockout animals. So, here in black, I'm showing you the wild-type neoantigen-specific CD8 T cell responses after sub-Q vaccination or IV vaccination. So in BATA3 knockout mice, which are lacking CDC1s, we can see that the responses are diminished both after sub-Q vaccination, but especially after IV vaccination. In CCR2 knockout or CCR2 DTR animals that receive diphtheria toxin, we can also see a drop here in green in the responses following both vaccination. And this suggests that there is a requirement for both CDC1s but also monocyte-derived dendritic cells. So to conclude, in this paper, we have described how the magnitude and quality of neoantigen-specific CD8 T cells are tunable by modifying the route and dose. Mechanistically, sub-Q vaccination resulted in prolonged antigen presentation, giving rise to terminally differentiated effector cells, whereas IV vaccination provided transient antigen presentation, resulting in less differentiated stem-like cells. This has important consequences on the anti-tumor capacity of these cells. I'd like to end off by thanking my mentor, Bob Cedar, as well as other members of the cancer team, our collaborators at Avidia, Andrew and Jeff, for providing the vaccines, our other collaborators at the NIH, as well as A-STAR in Singapore and in Mount Sinai. Thank you.